Hello and welcome everybody to the Variant Interpretation for Cancer Consortium General Call. We took a break over the summer and we are back. And we're really excited to have Dr. Jasmine Baker this morning from the Baylor College of Medicine. And she also does quite a bit of work with the ClinGen, kind of the hereditary cancer gene disease validity and relationships. So she's going to talk in detail about that work today, but we're really glad to have her, and this is really exciting developments in our field, I think, just to have more information at the gene level rather than at different levels, like a variant level. There's just a new utility for that that I get really excited about. So without further ado, I'm going to hand it off to Dr. Baker to get us started. Thank you so much for being here.
All right. Thank you, Beth, for the great introduction. So my name is Dr. Jasmine Baker. I am currently a staff scientist for the Clinical Genome Resource, and I'm also the coordinator for the ClinGen Hereditary Cancer GCEP. And today I'm going to present about the work that our GCEP has done on revisiting hereditary cancer gene disease relationships and some of the lessons that we have learned while completing this work. It's gonna click. Next slide. Oh. Disclosure. Before we begin, I want to disclose that I was an independent contractor for Johns Hopkins University in Open Cravat when this work was performed. For the scope of this presentation, I want everyone to remember that it's important that ClinGen's gene disease validity asks whether a germline variation in a specific gene causes a defined monogenic disease. It does not classify individual genetic variants, assess somatic oncogenicity, or evaluate therapeutic actionability. So throughout the presentation, I'm going to hit three major points. So I'm going to begin with what the ClinGen gene disease facility curations, what they actually curate, and how an assertion is defined. Then I'll explain why the hereditary cancer relationships were first assessed in their earlier effort and why they needed recuration. And finally, I'm going to discuss specifically the work with colorectal and polyposis and breast and ovarian cancer genes and what these results mean for clinical interpretation. So this first section is going to establish the foundation for the rest of the talk. So the key idea is that validity doesn't belong to a gene in isolation. It belongs to a precisely defined gene disease assertion. And once that assertion is clear, the evidence can be evaluated consistently. So a complete validity assertion includes the gene, the disease, the mode of inheritance, and the relevant germline context, which is the classification that you will have. ClinGen focuses on specified monogenic disease relationships rather than common risk or susceptibility alleles. So that is a really important point to make note of. So in short, what our ClinGen gene disease validity curation actually does can be kind of lumped into three major buckets. So first we have our assertion definition, and this is what happens during our pre-curation phase. It includes determining the disease entity and whether phenotype should be lumped or split with that disease entity. Next, our curators evaluate genetic and experimental evidence, including contradictory data. There is lastly an expert review that produces a classification of the relationship strength. And it's not a classification of an individual variant or judgment of tumor actionability, but just that particular gene disease relationship. So we determine which disease we're going to actually do the curation for, and then our curators go through all of the evidence through the literature, segregation data, et cetera, and then all that information is presented to our expert panels. For the recuration effort that I'm going to detail in the next section, I want to highlight what the objectives of the recuration effort were before we get any further, just so we're kind of all acclimated. So now we know what GDV is. The first objective that we had was we wanted to apply our updated standard operating procedures. So I'm sure everyone on this call or anyone rewatching this knows, once you get new data, once classifications and things are rewritten, you have to implement those new specifications. So that's what we wanted to do. And this QR code links to our most recent SOP updates. We also wanted to implement lumping and splitting. So lumping and splitting is a procedure that ClinGen has defined, and you can read more about it in the PMID listed below. And it goes through several steps that we'll go through in detail in the next slide for us to determine if we want to combine disease entities together, if we are doing a curation, or if we want to keep them separate. We also redefine disease entities and their dyadic naming with the help of our great friends Mondo with Monarch Archive. And I'll show you guys some of that today as well, and how you can see our ClinGen specific labels on their website. The next thing we wanted to do was to make sure that all of this evidence was entered into ClinGen's gene curation interface. So when the initial curations were completed for colorectal cancer, polyposis, and breast ovarian cancer, our gene curation interface did not exist. So we wanted to make sure that everything was updated and was properly into the system. And subsequently, if it's in the GCI that we have, it can be published as evidence on clinicalgenome.org, and then it's publicly available and anyone can go to the website and view the evidence. So now we're going to move into lumping and splitting. Lumping and splitting is the very first phase of the first step in pre-curation. So this, combined with literature searching, our curators are going to be answering yes or no to these questions. So they're going to see, is there an assertion for a distinct condition or isolated phenotype? Has it been made before? Maybe by OMIM or Mondo or a different group. Also, they're going to ask whether the conditions share a critical molecular mechanism. Next, they're going to look at phenotypic features that are observed between the families, suggestive of a distinct condition, or if it's a broad spectrum. And lastly, they're going to look at if there are differences in the inheritance pattern between the diseases. And based on how our lumping and splitting teeter-totters on our little triangle, you're either going to say your evidence favors a splitting of these diseases in relation to this gene, or you're going to lump them together. So we just discussed this pre-curation phase at the very top. Get my pointer. This pre-curation phase at the very top, where it is doing the lumping and splitting in the literature search, and this information goes into our gene tracker. And next we move on to our actual GCI. So once the pre-curations are presented to our expert panels, then the expert panels say, hey, we agree that this needs to be the proper lumping and splitting. This is the gene disease entity that needs to be curated. So next, our curators go through and they curate the genetic and the experimental evidence our system automatically assesses the evidence score based on what information is input by the curator. And it gives you an automated classification. So before this is approved, this classification is presented at our monthly our GSEP meetings. So expert review, the curator presents the information, our experts say yay or nay, or this is what we're seeing in the clinic, which is also very important to consider when you're doing these classifications. The final classification is approved, and then it is published to the ClinGen website. So the thing that a lot of people are very excited about, they go to our ClinGen website and they're looking for these classifications. So our classification system uses a graduated framework from no known disease relationship all the way through definitive. And disputed and refuted are used when credible contradictory evidence challenges the relationship. And each one of these labels communicates evidence strength for the exact assertion. So you want to make sure you're always reading this together with the gene, disease, and the inheritance definition. So this is an example that I have from clinicalgenome.org. So PALB2 was one of the genes that we did recurate. So it shows how the assertion is defined, and it gives us our disease term, which is PALB2 related cancer predisposition, which is a term we created with Mondo. It has the Mondo identifier under it. It tells you which expert panel curated this GDV. It tells you the classification, and then it also has the report and date. Okay. So this little lumping and splitting arrow here. So all of our GDVs have this little drop-down menu. And once you click this arrow, it'll pop open a screen. So it will show you all of the information that our curators put in from their pre-curation, where they did the initial literature curation and the lumping and splitting. So it will show you all of the OMIM information that was included and all the information that was excluded. So for this particular curation, breast cancer, pancreatic cancer, and breast ovarian were included, and Fanconi anemia was excluded. And then we also have a section where we include the notes where it says what's being characterized and specifically what happened in that lumping and splitting that made us exclude or include a certain diseases. I also want to highlight these green buttons. This is something that's not necessarily obvious to everyone initially. Once you click this green button of when the actual curation was completed and finalized, it will pop open another screen that gives you the gene, disease, mode of inheritance, expert panel. Is it replicated over time? It also gives you the ClinGen curation ID. So you can use that as well if you need that information to look it up in our allele registry. We have our it tells you which standard operating procedure was used. It also gives you the classification. And the next thing that's really important about this step, because it is a lot of it's curation, it's a lot of content. So we have these nice tabs here on the website where you can actually go through. It has our evidence summary statement, which lists the PMIDs, all the evidence that was curated and which SOP was included. And next we have where it goes to each individual tab. So you can click each tab and it'll show you the points at the top. And it will also show you rows of information in a table format where you can see each individual piece of evidence for genetic evidence, as well as the experimental evidence. If you go, it also has information for case control and case level segregation. So it'll list out the odds ratios. So typically when people are looking for information specifically for a GDV, this most likely is the information that they want in these individual tabs. So that's how you can get to these individual tabs. It also lists the experimental evidence, and I wanted to highlight before I move to the next section of the talk about the actual recuration efforts, that this little button here. So you can access our website through API, but if you're just looking for one piece of information on one GDV, you can go to the website, click that green button with the date that it was finalized, and you can navigate here. And this button will allow you to download the data in Excel format, text format, comma separated variable, or whatever format's easier for you to manipulate, for you to be able to access the data and have it downloaded. So if you need it, it's there. Okay. So why do recurations change people's conclusions? So a classification is a timestamp conclusion based on the standards, disease definitions, and evidence available at that time point. So when those inputs improve or change, the responsible thing for us as scientists and clinicians, if you are a clinician, I'm not, to say is whether or not the original work needs to be updated, and do we need to reassess this, and does the conclusion or classification still hold, and is it still relevant to what we're seeing? So the earlier hereditary cancer assessments created a really strong foundation. So 40 genes were evaluated in the colorectal and polyposis effort, and 31 genes were evaluated in the breast and ovarian cancer effort. I have the PMIDs for both of those original 2019 manuscripts here. If you guys would like to reference them, they're really good manuscripts. You should check them out. So since then, ClinGen's procedures, disease definitions that other people like Mondo and Omen have been updated. Available case control and cohort evidence has also evolved, and people are able to do larger studies now. So our recuration effort is asking, which of these original classifications remain supported under these updated conditions, and which ones need to be updated. So back to our lumping and splitting. So if you can reframe your mind to that system, that workflow we talked about, this is what we did for that PALB2 example during the pre-curation. So our curator, they did the literature, they presented everything. And we had OMIM, who had Fanconi anemia and breast cancer listed, and pancreatic cancer listed as disease entities for PALB2. Prior to our recurations, ClinGen said there was Fanconi anemia, breast cancer, carcinoma, and ovarian cancer, and hereditary non-polyposis colorectal cancer. So based on the information presented, the evidence favored lumping associated diseases into PALB2 related cancer predisposition. So it included breast, pancreatic, and ovarian cancer, while keeping Fanconi anemia and HMPPC split. I also have a really good reference here at the bottom. This is the manuscript on how to approach lumping and splitting and defining a disease by Dr. Thaxton and her group at UNC with ClinGen. It's really helpful if you're trying to get into gene disease validity curations and understanding how this lumping and splitting works in detail. So another big update and objective that we had for this recuration effort was dyadic naming and integrating Mondo. So if you go to Mondo's website or the ontology lookup service website, you can see that PALB2 related cancer predisposition is now a clearly defined disease concept that's supported by Mondo's terminology. It kind of helps us reduce the ambiguity when you're looking at historical labels, because sometimes they overlap, especially if you're looking at ontology trees. So these precise names that we have, where we have our gene name in the front with the related predisposition or disease in the back, kind of helps us get a framework and focus point on what you're actually discussing and what disease and gene you're actually looking at. This is the website that this particular screenshot is from. So it is from the version 4 of the Ontology Lookup Services website, and you can navigate to the different classes. This is a screenshot of the ontology tree. So if you navigate to that link or to Mondo or Ontology Lookup Service on the left side of the screen in the panel, they will have an ontology tree for whatever gene disease relationship that you are looking at. And this is a screenshot of what it looks like. So in collaboration with Mondo, we were able to get this updated. So now that our new disease entity is listed under hereditary neoplastic syndromes, and it's easier to locate in an ontology tree. So next, I'm going to move on to how we applied all these principles to colorectal cancer and polyposis. In this recuration, the updated evidence moved some relations upwards, while some were weakened and some were disputed. The range of outcomes is a really good point to why we need to kind of systematically reassess these things and recurate. So our team reassessed 46 colorectal cancer and polyposis gene disease pairs from the earlier 2019 evaluation. We needed to do this just as a reminder because our SOPs were updated and we have a gene curation interface now. There's also a lot more common cancer data now in regards to case control and population studies related to colorectal cancer. And also for rare polyposis, there was a lot of new evidence that strengthened rare polyposis disorders assertions that needed to be included in these classifications. So getting into the data. The updated evidence changed several of the classifications. So I have larger font on the left to show the updated classifications, and on the right, I have a visual to show you how the actual evidence types affected the curation change. So just going from left to right, for Axon 2, we went from moderate to definitive. And you can see on Axon 2, originally we were limited. So we had experimental evidence and then we had a lot more genetic evidence. But you can see that here for Axon 2, some of that experimental evidence, the bar is lower, but that is because that experimental evidence was refuted and not proven to still be valid. But there also was an increase in the amount of genetic evidence. So this particular curation was bumped up. So for ATM, we went from moderate to definitive. For NTHL1, from moderate to definitive, and so on and so on. But I do think it's important to also note that for our downgrades, we went from moderate to disputed and limited to disputed. So the takeaway from this slide is that not that any single gene is most important, is that the classification should follow the total evidence rather than remain anchored in its prior label or classification, especially as new evidence evolves and people are doing larger studies. So the overall colorectal and polyposis result is summarized in this flow diagram that we have here. So 20 of the relationships maintained their previous classification, but 26 of them were reclassified across upgrades, downgrades, disputed or refuted outcomes, and some of them were retired. So with retired, that means we've been looking at this assertion for a while and there has been no new evidence over a certain span of time, and it's just time to retire it. No one is even saying that this is a valid gene disease assertion anymore. So if you're trying to read this flow chart, you can see that before recuration, we started off with 15 definitive gene disease validity curations, and now we have 21. We don't have any strong anymore because it moved to definitive, which is nice, because that does help later on once you get to the clinical reporting side and people want to actually look and see which genes are associated with diseases. We only have one moderate now, when initially we started out with 7. We have way less limited. Our limited split in between definitive, and some of them moved back to limited, or either they were disputed, and a lot of them were just retired. And we do still have, for the no known disease relationship, those are retired now, and we do still have 2 refuted. So we'll see how that goes. The second recuration focused on breast and ovarian cancer relationships. So for this particular part of the effort, large case control data sets played a really important role in testing previously proposed associations. And these results show how broader and better powered evidence in studies can narrow down the set of relationships that are being considered credible. So in total, this part of the review assessed 54 breast and ovarian cancer relationships. 31 of those relationships were previously evaluated from that 2019 study, plus an additional 23 pairs. So these additional pairs came from assertions from a genetic lab test or that were in the literature. So that's how we got to that 54. So our curators applied these newer procedures that we've already talked about. They incorporated the new data from large case control studies, and they revisited the lumping and splitting and the naming. And as a result, we really got better supported and more precisely defined relationships for breast and ovarian cancer. So I have an example of one of those larger studies that I mentioned. So the Curious Consortium is an example of the scale of evidence that changed in the field. So these types of studies were able to provide more stable risk estimates, and it helped us distinguish well-supported associations from signals that weren't reproducible or either aren't reproducible currently based on the data and the people that were in the studies. So large studies can also, so this particular study is from the Breast Cancer Linkage Consortium. So these larger case control studies can also help us differentiate between the different types of cancer. Which was really nice because this study was it made it possible for us to examine breast cancer as more than one broad phenotype. The associations were differed by the estrogen receptor, if it was positive or negative. So that was very helpful when we were looking at ATM and BART1 specifically. You can see overall that the odds ratios were over here closer past this dash line, so past 1. But as you look here for positive and negative, you can see that there is a differential between ATM and BART1 in regards to the estrogen receptor being expressed or not in the types of breast cancer. This was also very important when we were assessing some of the newer literature that they have out and that is continuously coming out about triple negative breast cancer as well. So this flow, similar to the one we saw with colorectal cancer, it summarizes the recuration efforts. So we have 13 relationships that maintain their classification and 41 were reclassified, which is, it's a good bit. So we went from 10 definitive to 15. We don't have any moderates anymore. No strongs were in the original assessment at all. Our limiteds and our no known disease relationships, there was a lot of movement. So we moved to disputed, refuted, and retired. We don't have any moderates anymore. And our refuted is still refuted. I won't refute it. So okay. So 76% of those relationships were reclassified, which is really shows the power of having some of these larger case control studies and updating your curations and your classifications whenever you do get new information. So updating the evidence really made a huge difference for these breast cancer and ovarian cancer curations. So we have all this information. We're scientists. We love to read. We love to dig into the details and the weeds. But what do we do once we have all this information finally organized and it's available for people to look at and view? So we've seen how the validity of evidence can change and evolve over time. Now I'm going to connect that to clinical interpretation. So this particular table that I have here focuses on inclusion criteria for genes with various gene disease evidence levels. So this is from a manuscript by Bean et al. I believe. I can't recall the year at the moment, but this framework translates evidence strength into different test design decisions for diagnostic panels. Diagnostic panels generally require stronger established gene disease relationships because every reported gene can affect interpretation and follow-up when you're in that clinical setting. So in this particular table, I have in green. So if a gene disease relationship is determined to be definitive or strong, it can be included in your test results. If it is moderate, you see it gives this little caveat of a B here, which means you need to indicate in the report that the evidence for the disease association is still building. And variants are unlikely to be classified as above likely pathogenic. And you can see I have here in red for limited and no evidence. That means this shouldn't make the report at all. I can imagine that having some of these things like the limited and no evidence does give a lot of clinicians and patients a relief, less things to kind of focus on and have, anxiety about as you're looking at some of these diagnostic reports. So it's really important for resources like ours at ClinGen to keep our information updated because people do reference it and they do use our information as kind of an authority in the field of which gene disease validity relationships are actually accurate or not. So I do understand that this is a somatic group, typically, but where is the bridge for somatic? And, several habits I think can transfer directly from gene disease validity with germline curations to somatic, such as defining your exact assertion before you're scoring evidence, separating your mechanisms or your phenotypes that you're looking at, treating contradictory evidence as an interpretive input more instead of something that's more concrete, updating your conclusions whenever you have better cohorts or better data available, and making your level of confidence explicit. I figured maybe this might make a good discussion point if we still have time for questions, Beth, maybe if people are interested in that. So I have some conclusions on this slide, 3 conclusions. Hopefully this kind of wraps everything in. So first, validity really depends on both evolving evidence and a precise disease definition. Second, we have our 46 pairs of colorectal cancer polyposis genes. 26 are reclassified. That new evidence really strengthened the rare polyposis relationships because over time there was more data available because those diseases are more rare. So it took a little while to get that data available, but now those curations are more accurate. And for breast and ovarian, 41 were reclassified, and those large case controls really had a huge impact on how that data was interpreted and those classifications were updated. So recuration is really essential, and it should really be considered, I think, by a lot of groups whenever you are trying to be responsible in your interpretation so people can use them for other things down the pipeline. And that is the end of my presentation. So thank you guys for listening. I want to recognize the ClinGen Hereditary Cancer Gene Curation Expert Panel, all the collaborators, the experts who made this work possible, as well as our grants that are supported here. And you guys can visit any of the curated evidence on clinicalgenome.org. Thank you.
Thank you. That was wonderful. So kind of to kick off discussion, I was wondering if you could give some examples of how you see a lot of this work kind of translating to like a clinical laboratory setting or even like a research laboratory setting. You had mentioned something about like this being part of your like process in building diagnostic panels and things like that, but I'm wondering if you have other applications in mind as well.
So not necessarily a part of my process because I don't build diagnostic panels, but some of the labs, we do work with some of the their experts on calls and things. So they really lean heavy into having that information accurate so they can know what's plausible and what's not plausible. So outside of that, also having these classifications accurate and up to date, our website is publicly available. So outside of the panels, we also have clinicians, genetic counselors, and people who also go to our website for information. We also have people from other places and spaces that may not have access to all of the diagnostic testing. And that particular information with our gene disease validity curations that is actually able to separate things more easily into this is a disease, these are the phenotypes, and have it listed out. Those are some other use cases that people have been, well, I know they contacted me about personally. So yeah, as the coordinator.
Fantastic. And then maybe you mentioned this and I missed it, but is there a specific schedule that you plan to follow for recuration, or is it just
so we review all of our curations that are not definitive every 2 years. This was a larger effort because it was kind of the pivot point of we have the interface and a lot of curations needed to be updated at the same time. So that's why it became an effort more than the normal cycle of updating, every 2 years, but it's every 2 years for our curations.
And you said anything that's not definitive. And is there any value to recurating those definitive relationships?
I'm struggling with the word value. So there is value in having, if there is newer information, there is value in updating it if it does give you more information about the disease and the phenotypes available. So we do have sometimes where we do open up a definitive curation because there may be another GSEP that has some type of secondary information that is relevant to the disease spectrum, but it's not necessarily per se the cancer phenotype because we're with the hereditary cancer group. So maybe there is a prominent cardiovascular phenotype or something, so there are times where it does make sense to open that curation back up and update it. Because once again, people are viewing this information publicly. And if you're using it to develop testing panels and things, you want all of those phenotypes available there. And sometimes it makes sense to have it in one curation opposed to having it in two separate places on the website.
That makes a lot of sense. So even though that classification isn't going to change from definitive, nice to have that updated information in there. I'm going to open it up to the rest of the participants. If anybody has any questions or comments, feel free to either come off mute or post them in the chat. All right, I am hearing none, but yeah, did you have any other discussion points that you wanted to touch on? I know that there was one like couple slides previous that you maybe um you can go back like 2 slides. I think there was.
Yes.
Okay.
Yes. So there has been some discussion of somatic curation and gene disease ability, but I'm not sure where those conversations are. But I did think that maybe some of these topics would be great discussion points if people were open
Mm-hmm.
for discussion or if they had any thoughts on this particular topic. Um,
Yeah, well, those on this call, a lot of people on this call know that I've been working on like gene disease specific gene lists for different cancer types. And so I feel like this would be like a natural progression of like the gene disease validity work, but then we're kind of expanding that to something else. So I know that the CGC, the Cancer Genomics Consortium, has those lists and are revamping their website for those gene lists. So I'm wondering if we might be able to kind of, I don't know, it might be a good opportunity to kind of look at the criteria we're using, look at the
mhm.
criteria ClinGen is using, maybe align a little bit, and then if there's any, and then we can have, you know, joint standards on,
Yeah,
somatic curation at the gene level.
yeah, I think that would make sense. That would make sense.
Hmm.
Are there any other like experts on the call?
Yeah, I do see experts on this
I-
call.
I'll coordinate this call for you too, but any other experts who would like to give us some insight on this topic?
I think we think about these general ideas, broadly speaking, throughout various kinds of curation that are happening in civic. But I suspect that we've made different choices or think about them in slightly different ways. So some kind of direct contrasting would be really interesting to do. Um, can you elaborate a little bit more on what it means to make the level of confidence explicit?
So when I say make the level of confidence explicit, I mean having something similar to I'll pull it up on the slide so you can see um, a framework like this. Yeah, and it doesn't have to be these exact terms, but I do think something, to give people a sense of, I don't know, severity, I suppose, or seriousness of the information would be helpful, depending on who's looking at the information.
Yeah. Yeah, this is great. Um, and it's a little bit like a combination of evidence levels, evidence strength in civic, and then like kind of less structure, less formally described um, how we talk about in our guidelines or documentation about how you think about assembling multiple pieces of evidence when you're creating an assertion. Um, but those assertions are much more specific than
Mm,
they're not quite the same thing as gene disease validity. I think
okay.
so we have talked about trying to tackle gene disease validity more
Mm-hmm.
formally, and I think this is one of the things that we're thinking about for our next phase
Okay.
of funding. So I think when it comes to that point, it would be really smart of us to like maybe circle back to this and think about how we would collaborate to adapt this and not reinvent the wheel.
Yeah. It's a lot of
Yeah.
information to reinvent on. So, dog, I think a joint effort of some form would be easier for everyone involved, honestly, you know.
I don't see any reason why this couldn't be pretty closely applied to sort of, yeah, gene disease validity in a somatic cancer context. So, yeah.
I guess the only thing that comes to my mind that might be a challenge is that there are several genes in the cancer space that are just, they're studied so much, and you have so much data in there that like these numbers or like the independent studies may need to be adjusted for kind of like those KRAS, BRAF type genes, which I mean, I guess they're all going to be they're going to be definitive anyways, but in the rare tumor types where maybe they're not as important and they're still assayed, they might be
Yeah.
like overestimated. I guess that was like my one like hesitation here is everybody's
There's,
running a
yeah.
TSO 500 or something.
Yeah. Yeah, for like the major genes in the common cancer types. Yeah. Um, but there's probably some examples that could be found where there's like a rare alternate way of activating the same oncogenic pathway, and it's usually like one of these two or three genes, but like very rarely it's this fourth one. Yeah. But yeah, I agree. Yeah.
Cool.
And then as you got out into the rarer and more kind of like quirky cancers, it would probably like start to align closer and closer with the way this is being done. Yeah.
Yeah, that's what I think. Like,
Yeah.
um,
But
and
that would
yeah.
probably be not too difficult of a modification, or I don't know. Other than that, it might just kind of translate pretty nicely.
hmm Yeah, I think so. With the rare quirky cancers, like you were saying, like um, they typically have like diagnostic markers that you're looking for, and those would be your definitives. And then the other things, maybe in pathways or something, might kind of fall in the lower categories. But yeah, this is fascinating. I could talk about this for days, but
I mean, I think the overall principle, it's really unfortunate that we just haven't had the bandwidth to tackle this because I feel like the overall principle of all of this completely makes sense for understanding cancer drivers and which genes like truly are relevant. And because you do have this problem that there are some things that are obvious and really well established, but sort of formalizing that would still be valuable. But then there's this really long tail of rare cancers. And then there's also the problem that literally every gene in the entire human genome has been like called cancer related by someone. Like there's you can always find a paper, some lab somewhere is like making some claim. And that's really so when you do like literature searches, like you have a biological finding and you're trying to ask could this be interesting in cancer? You just always find something, and then it's sort of, but there's a wide range of like levels of evidence of support, and I think there's a lot of stuff that would fall into this limited, no known disease relationship, disputed, and sort of documenting some of the things that are in that category versus the things that actually rose to the moderate, strong, definitive would be really valuable.
Can I? Hi, everyone. It's a great presentation. Can I make a comment? I work with this somatic variants all the time for years, and I agree with you. And even with the limited somatic might have like different categories in terms of resistant versus the driver, and then how we're going to classify resistant mechanism, even in some genes, like a B2M mutations associated with less response with immune therapy. How we can, manage to classify gene disease relationship for some genes like that with those functions. So I think I mean, Beth is strong in this, but I think if it's the working group, we just discuss and brainstorm this, that would be great because when I was at Ambry, we did the gene disease relationship classification scheme is much simpler to work with. It was a number base. And then I sometimes use that still for somatic classifications of some of these genes, but still I'm confused when it comes to even the mechanism. Like when we look at the gene, even the type of variant is also important for the classifications. So very, very, very timely topic to discuss.
Yeah, wasn't even thinking of that. Would drug response or adverse drug response or any of those things be considered in scope for gene disease validity on the germline side of things, or is it really like quite scope to disease pathogenicity?
So that's more on the pharmacogenomics and form GK.
Yeah. There has been a little bit of a like, let's like focus the problem a little bit more, even though of course those other aspects are super important. Yeah.
No, they are, and the curators, they bring all the information, but you kind of have to keep the scope, focused on one thing and send the information.
Yeah. So this scheme is primarily used in the like, is this gene and variants in this gene pathogenic does it cause disease? Yeah.
Mm-hmm. Exactly.
Yeah.
Yeah, I like that thought though. Like you could even have, because you had those like paragraphs of different information in the different tabs on these. I could see there being, if we had this set up for somatic or something similar, having that kind of information about resistance or something kind of within that like tab structure, and having those genes kind of be definitive, but then like further clarifying why they're definitive based on the variant seen in those genes in the tab, kind of similar to what you did for these. So I could see that working. Cool. Great. Well, we are almost at time. Are there any closing thoughts or burning questions before we sign off?
Jasmine, I'm just making a note in our relevant Slack channel to reach out to you about when it comes time for our next phase planning. If we're going to pursue this topic seriously, we'll reach out to you if that's okay.
That's fine. I'll be around.
Great. Sounds good. Well, thank you again for presenting today. This was really great. We will post this to the YouTube channel. Oh, Andrea, I see your hand up. Are you?
Sorry, I wanted to clap, and it was a great presentation. Thank you.
Sounds good. Yes. Thank you. All right. Well, everybody have a great rest of your week, and we'll see you again in a couple weeks. All right. Bye.